Hello and welcome to part 2 of the Biology 6 Blood Lecture. In this second part, we're going to address two topics, blood types and blood transfusions. Now, to understand the blood types, we need to learn a little bit about the immune system. And the cells of the immune system, remember, are the white blood cells. Here in this slide of blood tissue, you can see 3 white blood cells, one there, and one there. Well, you might remember from earlier in this blood lecture, there are actually 5 types of white blood cells. We're not going to learn their names in this lecture, but there are 5 types of white blood cells. And all of them help you fight off pathogens. Pathogens are living things that can cause disease, like bacteria and viruses. Yeah, so your immune system, your white blood cells, helps you fight pathogens that have infected your body. And there are different ways in which the cells of the immune system helps you fight pathogens. But one of the most powerful ways that they do it is this. Some of these white blood cells make and secrete some Y-shaped proteins called antibodies. Let me click on a different view here. There's a blood vessel, and here you see a white blood cell going through it. But those Y-shaped proteins you see also going through the blood there are called antibodies. And antibodies are made by some of the white blood cells, and the antibodies move through your body fluids, including your blood. And how these antibodies protect you from pathogens is this. The antibodies can stick to the surface of a pathogen. And when they do, that leads to the pathogen being lysed, being torn apart. And so here in a moment, we're going to see a little cartoon of those antibodies attacking and lysing a pathogen. But before I get to that, I want to go on a slightly different topic and say this. How does your immune system figure out which are your own cells and which are the pathogen cells? You know, if like a pathogen, like let's say a bacteria gets inside of you, how does your immune system figure out which are the pathogen cells, which are the cells, of course, that it's supposed to attack, and which are your own cells, which are the cells that it's supposed to leave alone? Well, here's the way it works. Every cell, whether it's your cell or not, has molecules on the surface that the immune system interacts with, and we call these the antigens. If you look in the lecture handout, the antigens are molecules that the immune system interacts with. And when I say interacts with, essentially that means feels. The immune system has a way of essentially feeling the molecules on the surface of the cell. Now, what types of molecules are we talking here? Well, typically proteins and carbohydrates and lipids are the antigens on the surface of a cell that the immune system interacts with. Now, your immune system has essentially memorized all of your antigens. It knows what all of your antigens feel like, so to speak. And so we use the term self-antigens for that. Self-antigens are molecules that occur naturally in the body. Those are your own antigens. And so when the immune system sees that a cell has self-antigens on it, the immune system knows that's your own cell, and so the immune system does not attack cells that have self-antigens. But now let's say some other cell, let's say a pathogen like a bacteria has gotten inside your body. Well, that pathogen still has antigens. You know, it will have proteins and carbohydrates and lipids on its surface, but the thing is, bacteria have different proteins and different carbohydrates and different lipids from the ones on your cells. In other words, it still has antigens, but those aren't self-antigens. And we call these antigens that do not occur naturally in the body foreign antigens. So that phrase foreign antigen means any molecule that does not occur naturally in the body. And so if the immune system interacts with a cell and finds that the cell has foreign antigens, the immune system knows that cell doesn't belong in the body. It's an invading cell, and so the immune system attacks cells that have foreign antigens. And one of the most powerful weapons that the immune system uses for this are these antibodies. And so here they come. The antibodies stick to the foreign antigens on the pathogen cell, and this leads to lysis of the pathogen cell. Lysis, remember, means tearing apart or tearing open. And so there you see it. The immune system has basically shredded that pathogen cell using those antibodies. Okay, please keep all of those immune system concepts in mind, like self-antigens and foreign antigens, and that antibodies bind to cells with foreign antigens and lyse those cells that have the foreign antigens. Please keep all that stuff in mind because we're going to use it. We're going to use those concepts just here in a minute or two. But right now, we're going to switch to talking about blood types, which are sometimes called blood groups. And I'm sure you've heard of these, like maybe you know your own blood type, maybe just to make something up, maybe your blood type is O positive, but maybe somebody else in your family might be blood type A positive or AB negative. There are several types of blood, and we call these the blood types or the blood groups. But what are they? What makes one blood type different from another blood type? Well, what it comes down to this, it comes down to the antigens that are on the person's red blood cells. In other words, a person's blood type is set by what antigens they have on their red blood cells. And as it turns out, there are only three types of antigens, three major types of antigens that you find on red blood cells. They are called the A antigen, the B antigen, and the RH antigen. And that's it. These are essentially the only three antigens that you find on red blood cells, and so these are the antigens that set your blood type. Now, at first, you might think, so does that mean that there are only three blood types? But that's incorrect, and here's why that is incorrect. There are actually 8 blood types, not just 3. And the reason why you get more than 3 blood types is any given person doesn't necessarily have to have all 3 of these. So some people do have all 3 of these antigens on their red blood cells. So for some people, there are 3 self-antigens on red blood cells. But other people might have just 2 of them. So some people might have the A and the B and not the RH. And other people might have the B and the RH, but not the A. And other people might have the A and the RH and not the B. And some people might have just one of them. Some people might have the A, not the B, and not the RH. Other people might have the B and not the A and not the RH. And other people might have the RH and not the B and not the A. And some people might not have any of them. So as you can see, there's more than 3 possible combinations of having and not having each of the 3. And so, if you go through the mathematics and you try to figure out, all right, so how many blood types are there if everybody might have all of them or none of them, or any combination of 2 or 1 of them, and so you come out with 8 possible blood types. And I think they're easier to see if we just visually show them here. So those kind of pinkish looking things there are supposed to be red blood cells, and I put the antigens on their surface. So 8 blood types that are set by what antigens a person has on their red blood cells, or in other words, what are their self-antigens on their red blood cells. Now let's go through how the blood types are named. I mean, you see the pictures of the red blood cells here, but what do we call these blood types? Okay, so first of all, the naming works like this. You always say the A and or the B, depending on what they have. You always say the A or the B part first, and then if they have the RH antigen, you say positive, and if they don't have the RH antigen, you say negative. And let me go through a couple of examples, and you'll see what I mean. So this cell right here, whoever has this blood cell, they have the A antigen and they have the RH, so they would be blood type A positive. But a different person, if this was their blood cells, their red blood cells, they have only the A antigen and not the RH, so you would say they are RH negative. And there you have it. This blood cell it would be called A positive blood type, and if this is the type of blood cell a person has, we say that person is blood type A negative. And similarly over here, if this was a person's red blood cells, they have the B and the RH, so they would be B positive blood type. But this person right here, they only have the B, not the RH, so you would say that they are B negative blood type. This person right here has the A and the B and the RH, so remember you always say the A or the B first, A and or B first, followed by positive if they're RH, so this person is AB positive blood type. This person has the A and the B and not the RH, so they are AB negative. And the last 2 here, notice these last 2 don't have the A or the B, and they use kind of a special term for that. They say O. So when you're talking blood types, O means does not have A and does not have B. So both of, if these were this person here who has this type of red blood cell and this person here who have this type of red blood cell, they're both going to be O blood type, meaning they don't have the A and they don't have the B. But this person does have the RH, so they are O positive blood type. and this person here does not have the Rh, so they are O negative blood type. So there you have it. There are the 8 blood types, and it's worth your time to memorize this table, to be able to visually picture. If I say blood type AB positive, to be able to visually picture, a red blood cell that has the A and the B and the Rh. Or if I say blood type B negative as part of this lecture, be able to visually picture what the blood cells look like for that person of B negative blood type. It's just the red blood cells with just the B antigen. So, why is it so important to know a patient's blood type? Well, if I had to answer that question in a single word, I would say transfusions. And if I had to answer it in 2 words, I would say safe transfusions. So a transfusion means where blood is given to a patient. Why is that? Well, usually because the patient has lost some blood. Maybe the patient had an injury before they were brought into the hospital and they just lost a lot of their own blood from the injury, or maybe the patient is going to undergo an operation, and during the operation the patient will lose some blood and it needs to be replaced. So yeah, transfusion just means giving blood to the patient. Now, interestingly, you don't necessarily have to give the patient their own blood type for it to be a safe transfusion. It's always safe to give a patient blood that is the patient's own blood type, but you don't necessarily have to give them their own blood type for the transfusion. But you should never give a patient blood where the red blood cells in the transfusion have a foreign antigen to the patient. So if you give them a transfusion where the red blood cells have on their surface an antigen that is a foreign antigen to that patient, then that is very bad, and that could be harmful to the patient. That type of thing is called a transfusion mismatch, where you give a patient a transfusion where the red blood cells in the transfusion have a foreign antigen to the patient. Yeah, and it can potentially be fatal to the patient. Now, you might be wondering, well, why is it fatal to the patient if you give them this transfusion mismatch where the red blood cells have a foreign antigen to the patient? Well, let's imagine that this patient here is getting a transfusion mismatch by mistake, and let's imagine looking inside the patient's body. And so let's say this is one of the red blood cells that occur naturally in the patient's body. So this is one of the patient's own red blood cells. And let's say their red blood cell has some antigens. Let's for not worry if this is the A antigen or the B antigen or the Rh, but these are the patient's self-antigens on the patient's own red blood cell. And then let's say it's a transfusion mismatch and the hospital accidentally transfused some blood into the patient's body that had an antigen on its surface that's a foreign antigen to that patient, right? So that would be what we call a transfusion mismatch. And so here's why it's a problem. You tell me, what is the patient's immune system going to think that these red blood cells are that have just been put into the patient's body with the foreign antigen? It's going to think it's a pathogen, right? And the immune system, remember, is programmed to attack and destroy any cells that have foreign antigens on them because the immune system assumes that those are pathogens. And so, what you're seeing here is a transfusion mismatch because the patient has been given a transfusion where the red blood cells in the transfusion have a foreign antigen to the patient, and that activates the patient's immune system. The major way the immune system will attack these red blood cells that have the foreign antigen to the patient is with antibodies. Here you see these antibodies coming in. And remember what happens when the antibodies bind to the foreign antigens on a cell. The antibodies end up lysing the cell, and so you get a bunch of cellular debris, a bunch of torn up shreds of those red blood cells. And that's where the problem comes in, because that cellular debris is going to clog up the patient's blood vessels. So all over the patient's body, the blood vessels are going to get, clogged up with this cellular debris from the patient's antibodies attacking the red blood cells that had the foreign antigen. And that's always harmful to the patient when the patient's blood vessels get clogged with the cellular debris. It's always harmful, and about 20% of the time it's fatal to the patient. So anyway, the point of here is that transfusion mismatches are very dangerous to the patient and sometimes lethal. And we define them as whenever the patient receives a transfusion where the red blood cells in the transfusion have a foreign antigen to that patient. So this brings us to what I call the golden rule of all transfusions, and it's really the only rule of transfusions. Never give a patient blood that has a foreign red blood cell antigen to that patient. That's probably worth repeating. So never give a patient blood that has a foreign red blood cell antigen to that patient. Now, because transfusion mismatches can be so dangerous to patients, they very, very rarely happen in hospitals. Hospitals do at least 3 levels of checking, triple checking, before they give a patient a transfusion to make sure that it's not a transfusion mismatch. So basically what I'm saying is, if you or somebody you know is going into the hospital, it's very, very, very unlikely that they're going to get a transfusion mismatches because they triple check it before they do it. It's not impossible. You don't have to know this number that I'm about to say, but only about 1 in 10,000 hospital transfusions is a transfusion mismatch. But it does occur very, very rarely. Okay, so if you work in a hospital, and you're assigned to give a patient a blood transfusion, how do you check that it's a safe transfusion? How do you check that it's not a transfusion mismatch? Or in other words, saying the same question a different way, if a patient needs a transfusion, how do you figure out which of the 8 blood types is safe to give to the patient? Well, there's a method that's described in your lecture handout, a step-by-step method that will lead you. It's a way of figuring out which blood types are safe to give to the patient and which blood types are unsafe to give to the patient. And so let's go through this method of finding out the safe blood types to transfuse into a patient. Let's go through this method, and we'll have a sample patient just to make something up here. Let's imagine that this lady here who's going to get the transfusion is blood type A positive. So our goal is to figure out which of the 8 blood types can be safely given to her. Okay, yeah, so if you look in the lecture handout, it says that there's a method for finding the safe blood types for transfusion into a patient. And the first step, it says, is make a table of all the 8 blood types. And you should draw out the 8 blood types in your table, just like you see here. And I think you know how to do this. A positive blood type is the blood cells have the A antigen and the Rh antigen, and A negative blood type, draw a blood cell with just the A antigen. Yeah, so write out a table showing all 8 blood types and actually draw in the 8 red blood cell types, each with the correct antigens for its blood type. Okay, so that's step 1. Step 2 is this. To the left of your table, write in the patient's blood type, and we were told in this particular example that the patient is A positive, but don't just write that. Again, sketch out the patient's blood cell. So here's a red blood cell in the patient. She's A positive, so put the A antigen and the Rh antigen on this red blood cell that represents the patient's red blood cells. Good. And then the third step is this. Just below this patient blood type cell you've drawn, write in what the foreign antigens to that patient are. And what are those? Well, they're going to be all the antigens that the patient does not have. So remember that the 3 types of antigens that you can find in red blood cells are A antigen, B antigen, and Rh antigen. So the foreign antigens to a patient are the ones that the patient doesn't have. So just look at the patient's red blood cell and say, who's missing as far as antigens are concerned? And the answer in this case is B, right? The antigens that the patient doesn't have on the red blood cells is antigen B. And so that is the foreign antigen to this patient. So you put that in this box right there. Good. So that is step 3. Write out the foreign antigens to the patient, which in this case is just antigen B. And now you go back to your table of the 8 blood types there, and cross out any blood cells, any blood types that have the foreign antigen. So just go back to your table here and look at it cell by cell, and any cell that has the foreign antigen, just cross out that cell. Now in this example, the foreign antigen to the patient is B, so just cross out this cell and this cell and this cell and this cell. Those are the cells that have the foreign antigen to the patient. So those blood cells would be a transfusion mismatch if you gave them to the patient, right? And so the uncrossed out cells, the blood types that are not crossed out, are the safe ones to give to the patient, because those are the ones that don't have the foreign antigen to the patient. So getting back to our original picture here, for this A positive patient, it's safe to give her a blood transfusion that's A positive blood, and it's safe to give her a blood transfusion that's A negative blood, and it's safe to give her a blood transfusion that's O positive blood, and it's safe to give her a transfusion that is O negative blood. Good. So notice that most of these safe blood type transfusions are not her own blood type, right? Her own blood type is A positive, and certainly that's one of the safe ones, but these other ones, A negative, O positive, O negative, those are not her blood type. So what I'm saying is that some of the safe bloods that is given to the patient are not the patient's own blood type. So it's not true that the only safe blood to give to a patient is the patient's own blood type. The golden rule, remember, says don't give a patient blood that has a foreign antigen to the patient, and none of these have the foreign antigen. None of these have the B antigen, which is her foreign antigen. Okay, that was so much fun. Let's do another example just so you get the hang of it. So now let's pretend that this patient is blood type B negative, and let's go through these same steps again to figure out what the safe blood type she can be transfused with and what are the unsafe blood types that she can't be transfused with. So again, we start off making our picture table of the 8 blood types, the 8 red blood cell types here, and then to the left of the table you write what the patient's blood type is, which is B negative in this example, and then actually sketch out the patient's red blood cell, which is just the B antigen. Okay, and then the third step is below that, make a list of the antigens that are the foreign antigens to the patient. Okay, and remember, those are the ones that are not on the patient's red blood cells. So which ones are missing from this patient's red blood cells? Well, the A antigen and the RH antigen. So for this patient, the A antigen is a foreign antigen, and the RH is a foreign antigen. Those are the ones that are not self-antigens on the patient's red blood cells, right? Okay, and then once you've identified the foreign antigens to the patient, go back to your table of 8 blood cells and just cross out any that have either or both of these foreign antigens. So this has them, so we'll cross this one out, we'll cross this one out because it has the A antigen, we'll cross this one out because it has the RH antigen, that's a foreign antigen, we'll cross this one out because it has the A and the RH, which are foreign antigens, we'll cross this one out because it has the A antigen, which is a foreign antigen, we'll cross this one out because it has the RH, which is a foreign antigen. I think you get the idea. Just go through and cross out any and all blood cells that have the foreign antigens to the patient, like that. And so there it is. And so you can see when you go through this procedure for this particular patient, for the B negative patient, the blood types that are safe to give to the patient are B negative and O negative. And there you see it. There's the B negative patient, and the safe blood types that are safe to give to her are B negative blood and O negative blood. Let us do one last example just to practice this, but I'm going to sort of let you do this one on your own. Now let's say the patient is blood type O positive. And so what I'd like you to do is just stop the video right now, pause the video, and see if you can use this method that we just talked about to figure out which are the safe blood types to give to her. Anyway, that's your goal. Yeah, so pause the video and see if you can find out which are the safe blood types to give to this O positive patient. Okay. So did you give that a try? Let's work through the problem together and see what the safe blood types are for this O positive patient. So we'll make our table as usual, write out the 8 blood types and draw a picture of all the blood cells. To the left of the table, write the patient's blood type, O positive, and also sketch out the patient's blood cell. In this case, it's just a red blood cell that has only the RH antigen. And then below that, write the foreign antigens to this patient, which are the antigens that the patient doesn't have. And in this case, they are the A antigen and the B antigen, right? So those are the foreign antigens to this particular patient. And then go back over to your table of 8 blood cells and just cross out any that have the A antigen and cross out any that have the B antigen. And so in doing so, you would get this, leaving for the safe transfusions O positive and O negative. Yeah, so for this O positive patient, it's safe to give the patient O positive blood or O negative blood. So again, just to summarize what we've learned so far, it's not true that the only safe blood type to give to a patient is the patient's own blood type. In this case, the patient is O positive, and you can give them O positive or O negative. So you don't necessarily have to give the patient their own blood type, but you can't give the patient blood that has a foreign antigen to that patient. And so of the 8 blood types, these are the only 2 that don't have the foreign antigens for this patient, which are the A antigen and the B antigen. All right, well, I think hopefully you have the hang of how to figure out which blood types are safe to give to a patient and which ones are unsafe to give to a patient. Well, there are 2 special cases of patients and transfusion blood that I want to mention briefly. And here's the first kind of special case. If you did this method that we just talked about, you write out the table of the 8 blood types and then write the patient's blood type and then find the foreign antigens to that patient and then cross out the ones of the 8 blood type that have the foreign antigens to the patient. If you did all that for a patient who was blood type AB positive, you would find that you wouldn't cross out any of the 8 blood types. So patients who are AB positive blood type can receive all 8 blood types safely. None of the 8 blood types are unsafe to give to an A positive patient. And I tried to summarize that here, that for patients, for people who are A positive blood type, we call these people universal recipients. They can receive any of the 8 blood types safely. A positive people can. Oh, those lucky A positive people. And the other special case is this. If you did this method that we just talked about, where you draw the table of the eight blood types and then find out what the foreign antigens to the patient are and cross those blood cells off, if you did that for all types of patients, you would find that you never ever cross out the O negative blood. The O negative blood is never going to have a foreign antigen to any patient. In other words, O negative blood is always, always, always safe to give to any patient of any blood type. So, a bag of O negative blood can be safely transfused into any patient. So they sometimes call O negative blood the universal donor, because you can donate it to a patient. You can use it for a transfusion into a patient, and it's always safe. Or what they actually say is people who are O negative blood type, those people are universal donors, because those people who are O negative blood type can give their blood as a transfusion to any other person, and O negative blood is always going to be safe to transfuse into anybody. Okay, well, that brings us to the end of this lecture on the blood. We've covered a lot of topics. We've covered that the blood has 2 major parts, the plasma and the formed elements, which are the blood cells. We talked about that blood cells are generated in the red marrow of the bone tissues. We talked about hemostasis, how the blood leaking out of a torn blood vessel slows down and halts. We talked about what else? Anemia. We talked about the blood types and safe transfusions versus transfusion mismatches. Anyway, that's the end of this lecture. So as usual, I highly recommend that you now go do the review questions on this topic, and I will see you in the next lecture.